Raw of my wallow wrestling around and not like that. So I'm gonna put it back.
How's your weekend? How you doing? Okay, now I heard about that. I'm in a G-Com class and extra cut it out. Did you like it? It was 8 minutes to put it on, yeah.
No, they bring up issues of armor, so they're really
Right on, that's good. No, I work all weekend. I work maybe here Friday till Sunday night. I work intermittent homework when I can, and it's exhausting. I'm in school right now. Washington. I applied for it because I had bad back and ADHD. I was contracted, so they told me, hey, I did a neuropsych evaluation. I got accepted to the program, and based on my aptitudes, they told me to do the neuropsych evaluation. They assured me it wasn't passing. Do this and I do this evaluation. They say to them, don't contact me, leave me in the dark. And I contact me and say, hey, you did too. We can't help you. Not it was a pass fail. And then they tell me if I come back to school, they'll help me with housing. So I don't have to work while I'm in school, right? I get back here, I said, I'm in Michigan. Yes, absolutely. I get back and you're like, yeah, sorry. So, made it all under that name.
I'm sorry.
It is, so it just, all the time. This class isn't bad, but that genetics class was not one. You know what I mean?
Gotcha.
Yeah.
Gotcha. Well, happy Monday, everyone. Let's see, it's February. Help me out here. What's the
16?
date? February 16th. All right. So we're at the very end of chapter number 10. So that's where we're going to actually start off for today. And then I added some review questions. So I thought, well, instead of just reviewing at the beginning of class, because we're so close to the end of this chapter, we'll just kind of do a chapter review at the end of a few slides here. And then if you guys are up for it, we're going to move on to chapter 11. So that's the plan here. We do have our lab coming up tomorrow, so that'll be like a normal kind of lab there. Now, as we go along with these slides, I'll try and explain some of the things that we will be doing on Tuesday. But just prepare for more staining for Tuesday. So I want you all to look good, but don't wear your best clothing on Tuesdays. So I'm not going to wear my suit on Tuesday because of all the chemical stains and all that kind of stuff. All right, so we actually left off with this slide, but I know I was super rushed and I probably didn't explain it very well. So I thought, well, why not start with this one again? so if you're wondering, hey, I saw that before, you are not mistaken. At the very top of the slide, you're going to see the word phage. Let's remind ourselves, what do we mean by a phage? Yeah, no, Mondays are always rough. They're not easy on me either. So, if we can remember, what do we mean by a phage or a bacteriophage? Anyone?
It only affects the bacterial cells, is that what you said?
Yes, and what would be another word for phage or bacteriophage? Like, what kind of entity are we looking at here? Okay, something that's going to infect bacteria. I'm trying to give you a hint. There's a virus. That's right. These are viruses. Okay, good. Brady, you are correct. These are entities that, in this case, just infect bacteria. So those are the bacteriophages. Okay, and then we're going to see here with this kind of a what they call typing. What they mean there is, think of it as like a profile of what viruses infect a particular bacterium. So some bacteria will be susceptible to one set of viruses, but another species may be susceptible to a different set of viruses. So it's that kind of a pattern is what they're referring to as the typing. Okay, so how do you figure out what viruses a particular bacterium is susceptible to? Okay, let's look at these pictures on the right here. So here's our standard Petri dish. It has agar with food in it. And then on the top, they're adding in one bacterium. So in this case, it's Staphylococcus aureus, also known as Staph aureus for short. I don't know, have you noticed in science that they like to abbreviate, truncate words all the time, make things shorter? Okay, so there's, trying to imagine that Staphylococcus is growing all over the entire surface of this Petri dish. And then in the second panel, with this little pipette here, they're adding different bacteriophages. So each one of these little circles here represents a different virus. Okay? So lots of different viruses are being applied on top of the bacteria. And if the bacteria are susceptible, the virus will infect, multiply, and then cause those cells to rupture open. It's kind of like the original Alien movie. Do you remember that? When people were infected with the alien? Alien grows, people rupture open. Same concept here. But this time, it's the bacteria that are dying. And when the bacteria grow, they multiply and cause this haziness that you see here. It's kind of like this light gray color. But then when the cells lyse or split open, then you'll get this dark, clear. It's dark because the background is dark. So you get this clear zone that allows you to see through the Petri dish to the dark background. Okay, so remember, each of these little spots here represents a different virus. So then we can build a profile and say that our Staph aureus is susceptible to like this virus, this virus, this, and that, but not to these viruses over here. Okay, so that's a pattern. And then the idea is that you'll get a different pattern with a different bacterium. So in that way, you can distinguish, not only one species from another, but do you remember what we call the subspecies? Strains. You can look at differences in strains. Okay. So I'll put this one in the back of your memory banks here because we're going to do something similar at the end of this semester. So it's like, what's it like? 8 weeks from now. All right, any questions about this one? This is called phage typing. All right, we're going to look at a similar kind of setup here, but this time instead of applying viruses on top of bacteria, this time we're looking at paper discs that contain different antibiotics. And the profile that we're looking at here is called the antibiogram. So instead, again, it's a similar kind of concept, but instead of looking at susceptibility to viruses, different kind of viruses, now we're looking at differences in susceptibility to antibiotics. I guess the slide actually says antimicrobial medications. So that would be the antibiotic. But it doesn't have to be an antibiotic. You can actually use other kind of test solutions here, if you have an interest in that. So it could be different kinds of disinfectants or antiseptics you want to test to see how effective are they. You can do like a comparative analysis. You can test other kind of liquids. Let's say you're interested in different kind of beverages, like different kind of herbal teas, for example. Maybe you want to test, green tea versus eucalyptus tea versus lemon tea. Which ones, do they have antibiotic properties? Yes, they do. Or the essential oils, different kind of oils. You can test, a lot of different, substances using a similar kind of setup as what we see here. So again, you start off with a petri dish. You put bacteria over the entire surface of the dish. Okay, you see these lines? Those are lines created by the wire loop. The person went back and forth, back and forth, back and forth in order to cover the entire dish with bacteria. And we're only looking at one kind of bacteria in this test. And then using paper discs. Are you familiar with the hole punch? Okay, that's what you use. Make paper discs, and then you soak the discs in different solutions. So they could be the antibiotics. And then when you apply them on top of the bacteria, the liquid will diffuse out of the disc onto the surface of the petri dish. And then if the solution is antimicrobial, then it will kill the bacteria, cause them to lyse open, die, and then you'll get a clear zone so you can look through the dish and see the black background. But look over here. I don't see a clear zone like these discs. So what does it mean? What does this mean to you?
Doesn't work.
That's right, it doesn't kill bacteria, right? So this bacterium is not susceptible, but rather is resistant. It's resistant to that antimicrobial solution, which could be an antibiotic. So for example, let's just pretend these two dishes are two different strains. And let's just pretend that this colored disc contains the same antibiotic. This strain is resistant to this antibiotic. This strain is what? It's susceptible. This one is susceptible. Okay. And then you can see we're getting differences in the patterns. That's our antibiogram that we can use to distinguish this strain from that strain. Okay, what does it mean if these clear zones differ in size? Like this is pretty big, this one's pretty small. What does that mean? I guess this one may be a better comparison here. This one's bigger, this one is smaller. So what does that mean in terms of these two strains?
One's more effective.
One is more effective than the other, that's right. This one, because it's got a larger clear zone here, that means this strain is more susceptible than this one is. All right, any questions about that one? Is that pretty cool? Yeah. All right, I think we're getting close to the end of chapter 10 here. We kind of began looking at chapter 10 by looking at a phylogenetic tree of those three different domains. And do you remember what piece of DNA was used to construct that phylogenetic tree that showed us those three domains? So rather than looking at all of the DNA that's present in all these different species, scientists, they focused on one segment of DNA. Anyone remember what that one was? I know I had numbers and letters. I'll help you out here. The first part was 16, followed by the letter S, and then you finish it up.
rRNA.
That's right, rRNA, there you go. Ribosomal RNA. So we're looking at the DNA for that particular RNA. Okay. And then here's one more phylogeny, phylogenetic tree here. So using that kind of sequence information. So rather than looking at all of the organisms, that are on the planet, this time we're focusing on, like a subset of those organisms here. All right, let's see if we can't remind ourselves, what do all these lines indicate to us? All right, we're looking at, for example, on the very bottom here, 3 different bacterial species here. Okay. So we got, for example, Neisseria, alkalogenes, and nitros. Boy, this one's a tough one for me. Nitrosococcus. There we go. All right. So what does it mean by having the last two connected together and then joined with the Neisseria above it? What does that tell us about the alkalogenes and the nitrosococcus? What does that mean for those two?
Really? Someone?
Yeah, that tells us that the last two are more closely related to each other than they are to the Neisseria. Okay, everyone see how that one works? With the way that the lines are joined together? Okay. And then you can kind of, follow it, the lines to the left. That, for example, this last group of three are more closely related to, for example, the Pseudomonas that's above that than it is to the Rhodospirillum above that. Okay, so basically you're just kind of working, as your eyes travel to the left, look for points of intersection. The more points of intersections that you see as your eyes move to the left indicates greater similarities. Yeah. All right. Any questions about this one? This phylogenetic tree here. Okay. All right, let's end chapter 10 with some review questions here. All right, let's start off with, so this would be like questions over the entire chapter. So some of these things, you have to kind of remind yourselves about a little bit. So first question. Remember that this chapter is about taxonomies, and there's three parts to taxonomy. Who can give us the first part? Or who can give us any of the three parts?
Classify.
We have classification, so that's one of three. Okay. Identification, that's a second of the three. How about someone else for the third? Nomenclature. What does nomenclature mean? Naming. That's right. So we have classification, identification, nomenclature. Those are the three parts to taxonomy. Yeah. All right, good. And then we also learned about how things can be classified into one of three domains, and then we saw that big phylogenetic tree that indicated those three distinct parts. And you remember, what was the basis for separating species into one of three domains? Well, we actually saw two different phylogenetic trees. So they actually show you two different ways that you can build three different pieces of evidence for these three different domains. So one was actually using that gene that we just kind of refamiliarized ourselves with, that's known by the numbers and letters. What's the name of that gene? Starts with the number 16.
16S RNA.
That's right, the 16S rRNA, that's right. So that's one of the bases for the 3-domain system. And then, anyone remember what was the other one that they looked at that also ended up substantiating, supporting that same division of species into 3 domains? So think ribosome. So instead of just looking at the ribosomal RNA, you can also look at what part of the ribosome?
Amino acid.
The amino acids of the proteins, the ribosomal proteins. That's right. So those are two different things, right? The amino acids that make up the proteins of the ribosome and the ribosomal RNA sequences, those are two completely different things. And yet, when you look at those two pieces of evidence or information, for all the different known species on the planet, it ends up creating essentially the same thing. It classifies everything into 3 groupings or domains. Okay. Next question. What the heck is a dichotomous key? Anyone remember that one? Anyone? Dichotomous key.
Yeah, it's where you have 2 choices and you put a choice, and then you do 3 more choices, you get another choice. And it's easy.
There you go. It's a think of it as like a flow chart where you're given 2 choices, right? You have a question, and then 2 options, and then after you answer that first option, then you're presented with 2 more choices, right? And what is it? What can we use it for? For identification purposes, that's right. So if you have an unknown bacterium, you want to know what is it? What group does it belong into? We can use a dichotomous key, and that's basically what we're doing in the laboratory, right? Okay, question 4 is multiple parts here. So explain how the following can be used for identification purposes. So let's start off with the first topic here. Microscopic morphology. So how can bacteria differ in terms of their morphology?
Size, shape, color.
Size, shape, color, yep. So, Zach, were you going to add anything? Okay, good. All right, let's move on to the second one. Culture characteristics. What do we mean by culture in terms of microbiology? Are we talking about what language they're talking among themselves? What kind of foods are they eating? What kind of things they celebrate? No! What are we talking about here? Anybody?
Just the characteristics of what's in the petri dish, like the sample itself.
Yeah, so we're talking about how they're growing. So can you give some examples of what you've seen in the petri dish? What do you call those round circular blobs that are growing in your petri dishes? Colonies. And what do you call the edge of the colony? Like the outer portion of the colony. Margin, yeah. So they can differ in terms of margin. How else can colonies differ? Size. Color. Shape. Height, yeah. Elevation, yeah. There's a lot of different ways, right? Okay. All right, then we're looking at metabolic capabilities. Okay, what does that mean?
Oxygen.
Yeah, oxygen requirements. Yep. What else?
What they consume.
Yeah, what they could utilize for energy. Very good. Think about whether they ferment or not. And then the last one here deals with serology, serological characteristics. So what do we mean by that?
Proteins, polysaccharides, or prokaryotic cells, identifying markers, cell wall capsule, flagella, pillite.
Right, so we're talking about antigens that are recognized by antibodies. That's right. Yep. So different or antibodies will bind to different bacteria based on those external features that they may or may not have. Yep. Yep. Okay, number 5. What's the name of that gene again? 16 sRNA. Yeah, yeah. There you go. Boy, if you guys missed that on the next test, I don't know what to say. Okay, question number 6. We can distinguish different strains of the same species by the following items here. So we're talking about biochemical differences. Well, we kind of went through that with examples of metabolic capabilities, right? So like what kind of sugars they ferment, for example. Serological meaning what does this mean again? Antibody binding, antibodies, right? Okay, and then phages are viruses, yep. And the antibiogram, how would you describe that?
Bunch of dots.
Bunch of dots containing what? Antibiotics or other kind of solutions on paper discs applied on top of bacteria in that petri dish. Okay, so I don't know, when you were in high school and you went back to your residence, did your parents ever ask you, what did you learn today? What would you say? What did you learn in microbiology today? Nothing.
Pages are viruses.
Pages and viruses. There you go. Pages are viruses. All right. Yeah, I think it's always good to kind of what we call reflect. You know, kind of go back over. You know, we call it review, right? But also give yourself time. You know, let the information kind of sink in a little bit. All right. Now that we've reached the end of another chapter, I know some of you have already done this, but what do you need? What can you do next? You can upload your notes. What notes? What are we supposed to do? Notes? Yes, take notes. Upload your notes to the Blackboard site. Yep. And yeah, before our next exam. Okay, so I just want to remind people about uploading notes. Yeah. All right, any questions about chapter 10? All right, you want to start chapter 11 today or next time?
Next time.
You want to try next time? I'm okay with that too. All right, I'll hang out in case you have questions. Otherwise, we'll just start chapter 11 next time. So I'll see everyone tomorrow for lab.
Like, I thought APE. Yeah, I was like, wow.